and I'd like to talk about some of our recent work trying to develop T-cell-based therapies, immunotherapies, that might be applied to common epithelial cancers. So all immunotherapies can be divided into three major categories, and I have them here. The first would be non-specific stimulations of the immune system. These have been with us for a while. The first effective immunotherapy for any human cancer was the non-specific ability to stimulate effector cells with interleukin-2 with response rates of about 15% in patients with melanoma and renal cancer, with about 7% of those being complete durable regressions. That was first reported in 1985. Now, there are two ways to stimulate any physiologic system. One is to directly stimulate it. The other is to remove inhibitory factors, and that, in fact, is true for most physiologic systems in the body. And recently, there's been a good deal of interest in inhibiting these regulatory factors called checkpoint blockades that inhibit lymphocyte function. Anti-CTLA-4 has been approved by the Food and Drug Administration back in 2011, and just a few weeks ago, a second checkpoint modulator, anti-PD-1, has been approved. Now, these drugs have been shown to be quite effective in patients with melanoma. There's some activity in patients with renal cancer. About 15 to 17% of patients with smoking-induced lung cancers appear to respond to anti-PD-1. But the common epithelial cancers, called gastrointestinal cancers, for example, have shown no response to these checkpoint modulators. And so the activity is restricted largely to just a small number of histologies. Well, there's been a good deal of activity in cancer vaccines, literally over 1,000 papers published in animal models as well as in the human. We have no effective cancer vaccines today. The only approved cancer vaccine by the Food and Drug Administration is for prostate cancer, the Sapulacel-T. It appears to prolong survival by about 4 months, but there are no responses. Only 1 out of 300 patients showed an objective response. Only 8 of over 300 showed a PSA drop. Everybody progresses and everybody dies, but it does appear as if survival can be prolonged. There certainly are no cures. What I'm going to discuss this morning is this third aspect of immunotherapy, and that is the use of lymphocytes as a drug, in much the same way that we might use chemotherapy agents. By identifying T cells with anti-tumor activity and then passively transferring these activated immune cells that have an anti-tumor capacity, and we refer to that as adoptive immunotherapy. Now, some of the advantages of this kind of cell therapy are shown here. If you have a drug like a cell that can recognize a cancer, well, you can produce in the laboratory very large numbers. Well over 10 to the 11 lymphocytes are easy to grow, and you can select those kinds of cells for cells that have a very high avidity for recognition of tumor antigens. You can in vitro, because you have the cells in the test tube, potentially identify the exact cell subpopulations and the effector functions that are necessary to see cancer regression in vivo. And most importantly is this final point, and that is by taking immune cells out of the body, one has a chance to manipulate the host prior to the re-administration of these cells to provide an altered microenvironment for the transferred cells. In other words, a way to eliminate many of the inhibitory factors, T regulatory cells, myeloid-derived suppressor cells, by first wiping out the patient's own immune system transiently before giving the patient back their own anti-tumor cells. So all of these are autologous treatments. And so one of the early ways we did this is shown on this slide. We excise a tumor from a patient. We form multiple cultures, either starting from fragments or individual cells. We use in vitro assays of anti-tumor activity to identify those few cultures that have recognition of the cancer, and of course, these are likely to be a very low incidence in the repertoire. We then identify those cells, grow them to large numbers, and give them back to the same patient, but first following a transient lymphodepletion of that patient with cyclophosphamide and fludarabine at doses that eliminate all white cells for about 7 or 8 days before the body naturally recovers. Well, we published this several years ago. We now have a minimum 5-year follow-up on our earliest trials. We treated a series of 93 patients in three consecutive phase 2 trials. And I show you the results here with a minimum follow-up beyond 5 years. You can see we treated 93 patients with their own autologous anti-tumor cells. The objective response rate by RECIST criteria was 56%. And notice that 20 of the 93 patients, so 22%, had a complete regression of all metastatic disease. And when you have a complete regression with immunotherapy, and that's true for IL-2 as well, very rarely did those patients recur. Of these 20 patients, only a single patient recurred at 9 months, with all of the remaining patients in ongoing complete regression from 80 to 124 months. Now, a couple of examples just to make some points about how this works. First, because you're giving a very active drug that can immediately recognize the cancer, these responses can occur very quickly. You see, this patient that we first saw had multiple liver metastases. These were increasing in numbers. On day 0, we resected one of these metastatic deposits, identified anti-tumor cells, gave the cells on day 0, and you can see by day 34, virtually all of the tumor deposits are gone. There are still a few small remnants, but this patient underwent a complete regression that's now ongoing 11 years later of these lesions, as well as lesions that were present in the lung. So it can happen quickly. This patient had multiple subcutaneous tumors disappear in 12 days. There's no relationship between the bulk of disease and the likelihood of having a complete regression. And that shouldn't be a surprise. The immune system, when it recognizes an antigen, can destroy livers and lungs, and again, can destroy large tumor bulks as well. Now, this patient illustrates another point. He came to us from Israel with this terrible, very aggressive melanoma in the back of his scalp. He had had surgery, radiation therapy, chemotherapy, multiple excisions. We treated him on day 0, and you can see by day 22, everything is starting to turn black. But by 2 months later, the tumor started to regrow. We retreated the patient, and he underwent a complete regression of this disease, as well as multiple lung metastases. He's the only patient of the 20 complete responders who was treated more than once. And so all of the remaining patients that had responses and complete regressions that are ongoing beyond 5 years received a single treatment, because this is a living treatment. You're giving living cells. They expand thousands of fold inside the body after you give them. They retain their anti-tumor activity. And in fact, they live as long as we do. I mean, you get vaccinated against diphtheria as a child, you still have those immune cells 70 years later. Finally, this last patient that I'll show had multiple brain metastases, and I point this out simply to say that the brain is not a sanctuary site. Cells can infiltrate into the brain and cause regressions. And you can see these multiple metastatic deposits that disappeared over the course of 3 months. And so in this survival curve, if we look at the complete responders, you can see that one recurrence, but no other recurrences, and here we are out to 10 years with all of the patients having been followed for 5 years. The critical point of all of these studies that I'd like to make, and that is, if you give this tumor-infiltrating lymphocyte cells that we isolate from the tumor, which is a sink for anti-tumor cells, and select appropriate cells and administer them, you can eliminate the last cancer cell. These patients have ongoing complete responses up to 10 years and are very likely cured. Now, how do these anti-tumor lymphocytes destroy every last tumor cell? I should add as well that the responses and complete responses are completely independent of any prior treatment because the mechanism of this is so different. And as you can see in these patients that had either vemurafenib or IL-2 or ipilimumab or anti-PD-1, those numbers are small, response rates are the same. And so it looks like adoptive therapy can mediate complete, durable, likely curative regressions in patients with metastatic melanoma. But what's unique about melanoma? Patients with metastatic melanoma can respond to interleukin-2. They can respond to anti-CTLA-4, about a 10% response rate. They respond to anti-CD40. They respond to anti-PD-1. There's something about melanoma and to some extent renal cancer that appears quite unique. These are the only tumors that respond consistently to these immune modulators. It's the only tumor, melanoma, that can generate reproducibly large numbers of tumor-infiltrating lymphocytes with anti-tumor activity. And so there are two intertwining questions, the answers to which might give us clues on how to extend this treatment to non-melanoma patients. And that is, why is melanoma so uniquely immunogenic? And second, what do the TIL actually recognize that enables the in vivo destruction of the last cancer cell? Well, there have been multiple studies now where total exomic analysis has been done of tumors to look at the number of mutations. This is just one of those studies, and you can see them listed here. Each dot is a patient. This was published about a year and a half ago. And you can see that melanoma has more mutations on average, this is a log scale, than any other tumor histology, with the possible exception of smoking-induced lung cancers, and that might explain why anti-PD-1 has about a 17% response rate in patients with smoking-induced lung cancers. But all cancers have mutations, random mutations, but of lesser amounts. So we looked at a series of 33 consecutive patients with melanoma to look by exomic analysis at the number of somatic mutations that were present in those tumors that were not present in the genome. And you can see in our series of 33 patients, there was a median of about 234, an average of over 400. These are quite a few mutations compared to most other cancers that might have double-digit or 100 to 150 mutations. Well, we began studies to see if we couldn't mine this cancer exome, the cancer mutations, to identify whether there were mutations that were immunogenic. Now, for a mutation to be recognized by the immune system, it has to fulfill at least two criteria. The first is it has to be processed intracellularly to a 9 to 11 amino acid peptide, because T-cell receptors recognize small intracellular peptides that are brought to the surface of a cell and fit into the groove of an MHC molecule, which leads to the second criteria, and that is that depending on what MHC antigens that patient has, 6 class 1 and 6 class 2, the peptides that comprise that mutation have to fit in one of those patient's MHC molecules. And therefore, only some, in fact rare, mutations are actually going to be recognized by the immune system. So it's a critically important point. Any T-cell receptor that recognizes an antigen recognizes a small peptide in a groove of a surface MHC molecule. And that's true for both CD8 cells and CD4 cells. And so we developed a strategy that we published last year from mining all of the exomic mutations in a cancer to see if we can find some that were immunogenic. And I'll illustrate how we do this, but basically we take a whole exomic sequence of the tumor to identify all the non-synonymous mutations, that is, any mutation that creates a new amino acid. Most of the rest of the work is done sitting in front of a computer. In silico, then, once you know those mutations, we know the reference genome that's sequenced. So in silico, we can now list all of the 21 amino acid peptides that contain that mutation, flanked by the natural 10 amino acids on either side of that mutation. So by having this 21-mer or 25-mer with the mutation in the middle, That includes every possible 9 and 11 amino acid peptide containing the mutation. Well, in the first enumeration of this, we just use a prediction algorithm to identify whether any of these mutations are encompassed in peptides that can bind to a patient's MHC molecule. If there are some, we synthesize them and screen for in vivo recognition by TIL that cause a complete regression. So let me run through how we did this in a patient to provide perhaps a clearer example. The first time we actually did this was in this young boy, David, a 32-year-old male who had a fairly characteristic course of melanoma. He had excision of the disease. He had positive nodes. He didn't respond to interferon. He developed liver and brain metastases. He did not respond to high-dose IL-2. He had stereotactic radiosurgery to his brain and then an excision. And he was one of the 93 patients that we treated in the trial that I mentioned who had a complete regression of all of his disease, ongoing now at 9 years. Well, his exomic analysis showed that he had 595 non-synonymous mutations. So what we did, knowing what those mutations, the computer will then just tell us what protein that mutation came from. It will then tell us what are and that's this mutation here, this S, in place of that central amino acid. The computer then gives us the amino acids on either side of that mutation. So we now have, in this case, this 21-mer containing every possible 9 and 10 amino acid that contains that mutation. What we then do is have the computer just put them all together into a single string, and here it is. And now we ask whether or not in this string of amino acids, using prediction algorithms, are there any peptides that will fit on that patient's MHC molecule? We then synthesize those predicted peptides and use the patient's TIL that caused a complete regression to see if any of those peptides pulsed onto the patient's antigen-presenting cell will be recognized. And in fact, 2 were recognized. You can see this second predicted peptide was recognized in this co-culture assay with the patient's TIL that led to its complete regression, as was the 16th peptide. And this represented 2 tumor mutations, one a plextrin homology domain involved in microtubule formation, a second a protein phosphatase involved in glycogenesis in myotubes. And so these were 2 mutations that were recognized by this patient's TIL. A second example, this patient had multiple lung and metastases as well as bulky metastases in a mutation in an amputation stump. You see them pointed to with these yellow arrows. This patient that had multiple mutations that were present in the tumor, when we did this exomic analysis, predicted the peptides that would bind. You could see here, in this case, it was the 11th and the 23rd peptides that were predicted to bind to that patient's MHC molecule. And then once again, these were Gas7, involved in growth arrest, and House3, again, involved in microtubule formation. And so we can identify individual mutations that are recognized by TIL that are causing complete disappearance of cancer. Now, one of the problems with that approach is you have to predict sequences that can bind to a patient's MHC molecule, and there is such redundancy, especially in class 2 MHC molecules recognized by CD4 cells, that we don't have good prediction algorithms for many of those MHC molecules. Another problem that has prevented progress in cancer immunotherapy is that most tumors cannot be grown in culture. It's extraordinarily difficult to grow a colorectal cancer cell or a breast cancer cell or a prostate cancer cell in culture. And so we developed this technique, which was just published this year, that has multiple advantages compared to the previous one. There's no need to predict peptide binding to that patient's MHC molecule. You can, in one fell swoop, test all candidate peptides on any one of the MHC loci in that patient, and you don't need the tumor cell line at all. So what do? The first thing you do is you identify the exomic mutations, as we did before. And now what we do is we have the computer again tell us what the amino acids are on either side of that mutation. But instead of doing a prediction, what we then do is clone each one of the DNA mini-genes that encode that 21-mer, in this case a 25-mer. We clone that into an expression plasmid. We make RNA. We transfect it into the patient's own autologous antigen-presenting cells. So we put every mutation, every mini-gene, into the patient's own antigen-presenting cells to see whether or not the patient's own antigen-presenting cells will present that peptide by testing those antigen-presenting cells in a co-culture assay with the patient's T cells that mediated a complete regression. And so what we do to actually do this is we string each of these individual mini-genes encompassing 25 amino acids into what we call a tandem minigene that we then put into the patient's own antigen-presenting cells and then simply interrogate with the TIL that we used to treat the patient. And so here's an example of this patient with melanoma who had a small number of mutations. He had 71 non-synonymous mutations. Well, we made 12 tandem mini-genes, each containing about 6 individual genes. And here you see these 12 tandem mini-genes. And here's an example of one. This encoding 25 amino acids, this with the mutation in the middle, this another 25, and so on. Well, when we test against the patient's TIL lymphocytes that we gave to the patient, you can see only this first tandem mini-gene is recognized. None of the others are recognized. This one happens to be the autologous melanoma, which is recognized. Well, this tandem mini-gene encodes 6 different mutated peptides. To determine which is the gene that actually represents the mutation recognized by the patient's immune system, what we simply do is one by one mutate that mutation back to wild type for each one of the 6 genes in turn. So you can see here in this co-culture assay, when all 6 of these mini-genes have the mutation, it's recognized. When you now take this first gene and revert it to wild type, you can see the tandem mini-gene is still recognized. But when we take this second gene and revert it to wild type, all reactivity is gone, indicating that it's this gene, this KIF2C, that was the tumor antigen recognized by these TIL. And in fact, it turns out to be a member of the kinesin family, 2C. It's involved in the mitotic spindle. Well, shockingly, we've done this now in 21 patients and identified 45 different mutations. And every mutation was different. We've never seen a duplicate mutation recognized by the immune system. So in these 21 patients, we identified 45 mutations, but none were shared. Each were unique to the somatic mutation present in just that patient's cancer. And so it looks like, because we identified these mutations using T cells that could cause complete regressions, it looks like adoptive cell therapy mediates complete, durable, and likely curative regressions of metastatic melanoma based on the recognition of these immunogenic cancer mutations, because we see no side effects in these patients. There's no on-target off-tumor toxicity because we're targeting, again, mutations present only in that cancer. And in fact, these individual mutations are very likely the targets of interleukin-2, of anti-CTLA-4, and anti-PD-1, which can also cause regressions, but in much smaller percentage of patients. Well, having made this conjecture, can we utilize this approach to treat common epithelial cancers by targeting the unique immunogenic mutations that are expressed by each individual tumor? Might this be a way to use immunotherapy for the treatment of, or at least attempted treatment of any cancer type? So we chose for the next series of studies gastrointestinal cancers because these are among the kinds of cancers like prostate cancer and many other of the epithelial cancer that do not respond to IL-2 or the checkpoint modulators. And you can see, although they don't have as many mutations as melanoma, which is all the way to the right, I've boxed here in red the gastrointestinal cancers. They have maybe 1/10 the number of mutations on average as patients with melanoma. And so the first patient that we attempted this with was a patient that we reported in Science about 2 months ago. A 45-year-old woman who had a typical history of a patient with a metastatic GI cancer. She had a metastatic cholangiocarcinoma, bile duct cancer. She had had a right hepatectomy for this. She subsequently developed multiple lung and liver metastases, was given cisplatin, gemcitabine, didn't respond, taxotere, didn't respond. Came to us in March of 2012. We resected a lung lesion and isolated her reactive TIL and infused them. And she had just a sort of a stabilization of disease. She certainly did not have a RECIST response when she got her bulk TIL cells. Well, we did our whole exomic sequencing of one of these lung tumors, and she had 26 mutations, and here they are all listed sequentially. We know the exact protein that they came from and the exact amino acid that was mutated. So with these 26 mutations, what we did is we now made 3 tandem minigenes. We made a 25-mer that encompassed every one of the mutations. We then strung those minigenes together in a tandem minigene and transfected it, electroporated it into the patient's own antigen-presenting cells to see if her antigen-presenting cells would actually present anything to the TIL that really didn't work but caused a minor stabilization. So here are the 3 tandem minigenes, each encoding multiple of these 26 mutations to encompass all of them. And by putting them in our own antigen-presenting cells, we're looking at reactivity on any one of her MHC molecules. And sure enough, tandem minigene 1 showed reactivity and not tandem minigene 2 or 3. We could see that in an ELISpot assay, but also because of upregulation of OX40. Now that's something that CD4 cells do when their T-cell receptors interact with antigens. CD8 cells tend to upregulate 4-1BB. And so this suggested that she had CD4 cells, not CD8 killer cells, that were reacting with her tumor. Well, we then reverted each one of these each one of these genes that were encompassed in the tandem minigene back to wild type. And as you can see here, when we reverted these to wild type, nothing happened. We still had reactivity with the TIL, but when we converted this one, this ERB-B2IP back to wild type, the TIL no longer recognized it, thereby identifying this as the immunogenic mutation recognized by this patient's TIL cells. It's a tumor suppressor that binds to ERB-B2. It attenuates downstream RAS and ERK signaling, and is very likely a driver mutation. Well, we purified these ERB-B2IP cells so that almost 90% of all of the cells in this particular subculture were reactive with the ERB-B2 mutation, and we used that to treat the patient. And she's now undergone an ongoing regression. Her last follow-up was at 12 months, about 2 weeks ago. And you can see she has an ongoing regression now of this metastatic cholangiocarcinoma in both the lung and the liver. And so by targeting this individual mutation, we could mediate a dramatic response in this chemotherapy refractory patients. And you can see here the 6-month X-ray. I don't have one yet at the 12-month. I haven't made one yet, but you can see these large tumors which have shrunk dramatically. This tumor has disappeared. This one is shrinking, and at the 12-month period, these tumors are still active, and the patient is still showing us ongoing tumor regression. Well, we did this in a second patient, again, a patient who came to us with an ECOG status probably of 2 to 3, had been through a lot, This young woman had been diagnosed with a rectal cancer. It had chemotherapy and abdominoperineal resection, mets to the lung resected, a sacrectomy, bladder implants, multiple chemotherapies. Came to us finally in August of 2013 with progressive disease. We resected a liver lesion to grow TIL, but she died before we could actually give her these cells. Would have been much nicer, of course, had we been able to obtain her tumor earlier in her course. But it served as material to ask whether or not this patient really did have immunogenic mutations that could be recognized by her immune system. Well, she had 155 mutations in this rectal cancer. And what we did here is we separated CD8 and CD4 cells and tested them separately, utilizing multiple tandem minigenes. In this case, we made 10 tandem minigenes so we could encompass all of the 155 mutations. These tandem minigenes containing 4 to 16 genes each to look for reactivity of CD8 cells and CD4 cells. And notice here, this one particular fragment among many fragments tested recognized tandem minigene 2. So the CD8 cells recognized tandem minigene 2, but the CD4 cells recognized tandem minigene 5, here in green. So it looked like there were several antigens that this patient's immune system were recognizing. We did something a little different. Instead of just reverting them back to normal, the other thing you can do is just synthesize the 25-mers, pulse them onto the patient's antigen-presenting cells, and just see which gene is responsible. And as you can see here, when we looked at the CD4 cells, there were 2 different peptides that were recognized, this CHD6 and this GPD2 that were recognized by her TIL. The first was a chromodomain helicase that's involved in chromatin remodeling, and a glycerol 3-phosphate dehydrogenase that's involved in mitochondrial metabolism. We did this for her CD8 cells. Again, this tandem minigene 2 recognized just a single antigen. In this case, it was NUP98, a nucleoporin that's involved in the nucleopore complex. So this patient had at least 3 potentially targetable mutations by her TIL. But again, she was not treated because she died before we could accumulate this information. It takes about 8 weeks from the time you have the tumor in place to do the exomic sequencing. During that time, you're growing TIL, and then to do the bioinformatics, to do the synthesis of the minigenes and the final testing. So it can all be done in about 7 or 8 weeks. Well, how common is this phenomenon for the GI cancers? That is, how often do you find mutations that are recognized by the patient's immune system so they can potentially be used as targets for therapy? We can not only identify the cell, but because these cells have undergone a good deal of proliferation and are somewhat exhausted, it's quite easy when you have the cells to identify the sequence of the alpha and beta chains, which one can do really in the course of a few days by deep sequencing or 5' race. You can put them into a retrovirus and then show that that T cell receptor actually does recognize the mutation, as we did in this case. And so we've now tested 8 patients with metastatic colorectal cancers with a variety of histologies, cholangiocarcinomas, rectal cancers, esophageal cancer, colon cancers. And in 7 of the 8 patients, we've been able to identify immunogenic mutations recognized by the patient's TIL. We have not treated these patients yet, but at least we have the realization that patients have either one or sometimes multiple immunogenic mutations that we might use as targets for immunotherapy. And that brings you up to date with where we are now, aggressively taking patients with virtually any GI cancer diagnosis, but we're trying to get them a little earlier in their course, so we have this 8 weeks available to develop the cells or the T cell receptors that we might use for treatment. And so I would conjecture that the identification and selective targeting of mutated antigens that a unique to each patient's cancer may be a means to apply cell transfer immunotherapy to patients with common epithelial cancers or cancers with any large number of mutations. So this, in some sense, is the ultimate personalized therapy, because we're identifying a target that's only in that patient's cancer, and we're using that patient's cells to treat that patient. We basically have to create a new drug for every patient. It's a daunting prospect, but at least it holds the possibilities now for developing immunotherapies for more common kinds of cancers. Well, we've talked about mutations that are unique to each individual cancer, and I'll just finish by talking about two other classes of antigens that one can attack utilizing cell transfer therapies. The biggest problem in all cancer therapy, of course, is targeting the cancer and not normal tissue. Not a problem if the normal tissue is disposable and you can do without it. And so one prominent example of this second class of antigens are antigens that are expressed on cancers and non-essential normal tissues. So we've raised T-cell receptors against thyroglobulin, present on some thyroid cancers and a disposable thyroid, but the most attention has been given to CD19, which is expressed on almost all B-cell lymphomas and leukemias. And if wiping out normal B cells, which also express CD19 as a side effect, well, we can live without B cells by giving immunoglobulin transfusions. And so what we do here is create cells in the laboratory by genetically introducing either conventional T-cell receptors that have an alpha and a beta chain, and that's what TIL have used to recognize, that's what all T cells use to recognize antigens, alpha beta chains that have intracellular signaling chains that can activate the lymphocyte. But about 12 years ago, Zelig Ashar and his team at the Weizmann Institute developed what's called a chimeric antigen receptor by taking the heavy and light chains of an antibody, the variable regions, combining them into a single chain and attaching them to the intracellular signaling chains of a lymphocyte. So now you can create in the laboratory a lymphocyte that actually recognizes on the basis of an antibody. And so that's what was done to attempt to target CD19. Jim Kokendorfer, working with us, made this construct against an anti-CD19 using an anti-CD19 antibody by making a single chain antibody, connecting it to CD28 and zeta, putting it in a retrovirus. We made that retrovirus under GMP and then used it to treat patients. And the first patient that ever received an autologous T cell that targeted CD19 was this patient, who was a 48-year-old male who had an indolent type lymphoma. We'll talk about the more aggressive lymphomas in a moment. He had this diagnosed back in 2002. Typical course, chemotherapy. These tend to be responsive, but he recurred. They're not curable, of course. He received an atypic vaccine, and then ipilimumab, and then more EPOCH chemotherapy, and then recurred. And he came to us with major bulk disease. I'll show you his x-rays. He received his own autologous cells that were transduced with a retrovirus that enabled his cells to express this anti-CD19 CAR, and the patient underwent a dramatic regression and remains now progression-free by now over 5 years later. So he had this bulky disease in his mediastinum. You can see here, again, we see no relationship when you get an immunotherapy response to bulk of disease. You can see these large nodes surrounding the aorta and vena cava, these huge iliac lymph nodes, which went away. He had his marrow that was full of tumor that also cleared. And the price that you pay is this elimination of normal B cells, which went down to 0 and stayed that way for about 8 months, at a time when all of the other cells, T cells and NK cells, were recovering normally about 8 days after the lymphodepleting regimen. And so we treated 6 patients with indolent B-cell malignancies. They had all had multiple prior therapies. They were chemotherapy refractory. 4 of these 6 patients have ongoing complete regressions and 1 ongoing partial regression. But these are the indolent lymphomas. The large cell lymphomas present a more difficult challenge. We've now treated 9 patients with large cell lymphomas, and all but one of these were refractory to their last chemotherapy regimen. And as you can see, we have here 5 ongoing complete regressions and some partial regressions, and we're working hard to improve upon the treatment of these chemotherapy refractory patients utilizing this immunotherapy, as are other groups at the University of Pennsylvania now, at Memorial Sloan Kettering and the University of Washington as well. But to give you some examples, again, as you see from this PET scan, every one of those black dots represent a tumor which disappeared. This patient had his whole right chest replaced by lymphoma. You see the PET scan on the bottom that cleared, as you can see on the right. This patient had multiple large liver metastatic deposits that disappeared as well. So this anti-CD19 gene therapy can be quite effective for patients with lymphoma. The problem with the more widespread application of CARs is that you need an antigen, you need a molecule on the cell surface that an antibody can recognize. And unfortunately, in 40 years now almost of studies of monoclonal antibodies, we haven't been able to find hardly a single monoclonal antibody that recognizes a cell surface determinant unique to cancer. And the antibodies that are in use tend to interact with growth factors which inhibit the growth of those cells. But there don't appear, at least people haven't yet identified good monoclonals that can recognize cell surface determinants useful for killing these tumor cells directly. Now finally, there is a class of shared antigens, first described by Thierry Boon in Belgium in 1990, that are called cancer testis antigens. These are expressed during fetal development, but most of their expression is lost in adult tissues, with the exception of male germ cells, which don't express class 1, so they're immunologically inert. And the family that we've studied are the NY-ESO-1 and the MAGE family of antigens. There are well over 100 of these cancer testis antigens. And if you use RT-PCR, you can see with the NY-ESO-1 here in green and MAGE in blue, you can see expression on a fair number of these different tumor types. But if you use immunohistochemistry, the amount of expression, which is what you need to put a peptide on the surface, is about a third of these levels. Well, we made T-cell receptors, genes encoding T-cell receptors against NY-ESO-1 that could recognize this on melanomas as well as on synovial cell sarcomas, which have a translocation in the X chromosome and have high expression of NY-ESO-1. And you can see in melanoma, we got a 50% response rate using now, quite astonishingly, a patient's normal cells that we just put in this T-cell receptor, and now this cell, when grown and administered, can mediate regression. You can see a 50% objective regression rate, including 3 complete regressions ongoing beyond 2 years. And in synovial cell sarcomas, patients highly refractory, a median of 4 different chemotherapy regimens, a 67% response rate. Many of them are short, but some of them can be quite durable as well. And when you give a T cell, whether it's genetically engineered to express the antigen or not, you see the same kind of responses that you see when you use TIL against mutations. And so in this patient with synovial cell sarcomas, as you can see, these multiple lung metastases, this is of course what kills patients with sarcomas, as the growing tumors replace the ability of the lung to oxygenate. Almost a complete regression in this patient out at 18 months. There's still slight remnant, which is why we call her a partial responder. This large primary lesion, which had destroyed the hip joint here, seen on the left, is undergoing an ongoing regression as well, with beginning recalcification of the hip joint. And finally, this patient with synovial sarcoma again. So it's not just melanoma. What's unique about melanoma is the fact that it naturally creates T cells. If you can generate a T cell that will recognize a tumor, there's no difference in different tumor susceptibility to T cell damage. and it works in melanoma. Well, this, in this penultimate slide, is the program we now have in progress in the surgery branch of the National Cancer Institute. We are looking at a whole variety of different cell transfer therapies utilizing genetic modification of cells, targeting a wide variety of different antigens. We've explored this chondroitin sulfate antigen that Dr. Ferroni has so elegantly described. We're targeting the CGFR variant 3 receptor present on about 40% of glioblastomas. But there's a good deal of difficulty in identifying cell surface proteins that are targetable, with the exception of these isolated examples of mesothelin and this chondroitin sulfate. So in these brief remarks, I've tried to discuss the following points, and that is that cell transfer therapy can mediate durable regressions in patients with metastatic cancer refractory to other treatments, that autologous peripheral lymphocytes can be genetically modified to express anti-tumor T-cell receptors that can mediate cancer regression, and perhaps most interestingly, the identification and targeting of mutations unique to each cancer has the potential to not only improve upon the therapy for melanoma, but potentially extend this cell transfer therapy to patients with common epithelial cancers, and that's the subject right now of our most vigorous efforts. Well, thank you for your very kind attention.
I can't help but ask this question. Do you think there might be a limited repertoire of really good antigens that mediate the immune response to several of these different therapies that you're talking about, and that by sequencing patients prior to therapy, you could say this patient is going to be a responder, this one won't? I mean, that would really change the ballgame for us.
Well, again, most mutations are not immunogenic because they have to be processed and they have to match that patient's MHC molecule. We studied 21 patients. We found 45 mutations. There wasn't a single duplication among the patients. And that was within the melanoma histology. So we've also seen no duplications in the GI cancers in the 7 in which we found the mutations. So this is a whole different concept. We're not trying to find mutations that can be targetable by drugs, and there are plenty of gene libraries for which people are trying to do that. The problem is there's just not very many targetable agents that can cure patients. Here it's something completely different. Here we're just trying to recognize a mutation that can be a target of immune attack. And I think it's going to be very hard to just find the mutations and predict them without actually doing this kind of in vitro test.
May I make a comment? I agree with you. We don't have many targets we can recognize with antibodies. But the major advantage of the CAR approach is that one CAR can be utilized with a large number of patients. So just the antigens which we are investigating is expressed on 80% of patients with melanoma, at least. So that means if we develop the right CAR, we don't have to go to analyze each individual patient for expression of the right target. And
well,
I believe that is very important from a practical viewpoint, because as you showed, many times you are racing with disease progression, and you lost a patient while you were identifying the right mutations.
Well, I agree that it would be wonderful if we had a common mutation that the CARs could recognize. The problem is they just don't exist. I've told you about some of our successes. I haven't told you about some of our catastrophes. When we tried to target antigens that were present even in tiny amounts on normal tissue. And so when we targeted CEA, for example, which is way overexpressed in many colon cancers compared to the tiny amounts of normal tissue. We completely eliminated, wiped out the colonic epithelium of 3 patients. One of them had a really good regression of liver metastases, but it took 2 to 3 weeks for those patients to recover their colonic mucosa, and during that time there was terrible diarrhea and bleeding and so on. If you target a melanocyte antigen, there's a tiny layer of melanocyte-expressing cells in the stria vascularis of the cochlea, and T cells will find them. That's why we're protected against viral diseases. We have no drugs. Virus puts an antigen on the surface, a processed peptide, and lymphocytes that are circulating infiltrate in organs. When they see those, they stop, they expand, they have their effector function, and then they return to the circulation via the thoracic duct and continue to patrol the body. There are no cell surface determinants that I'm aware of that are unique to cancer, with the possible exception of this EGFR variant 3 mutation in patients with glioblastoma. We, as you know, have explored the chondroitin sulfate. We find it on normal cells. We're studying mesothelin, which is present on normal pleura and pericardium, and when that's been used, severe chest pain has been a limiting complication. So I agree. It would be wonderful to have one cell type that could, one target that could treat many patients, but so far nobody has found them.
You showed one patient from Israel that had a recurrence, and then you were able to treat that individual again with a complete response. Did you re-isolate lymphocytes or TILs from the recurrence, or did you use the original one?
Original cells. So for some reason in that one patient, all the other patients that had complete regressions received one treatment, but for some reason that patient, we don't quite understand it, did recur at 2 months, but with a tiny volume, and then by retreating him, he's undergone a complete regression that's ongoing now. But it was the same cells.
Yeah, I would have thought that antigen would go away, but it did not. That's interesting.
Apparently not, yeah.
I was curious about the heterogeneity of some of these things. So if a patient has multiple immune responsive peptides, do you have to give back the TILs recognizing the 2 or 3 or 4 mutations, or would one be enough? Or do you know what I'm trying to say?
I surely do.
Yeah.
It's entirely possible that attacking a somatic mutation, which is not essential for the malignant phenotype, could get downregulated. And so what we do know, however, is that in 20 out of 93 patients with melanoma, the last cancer cell disappeared. That is, whatever the TIL were recognizing could not be downregulated and did eliminate the cancer. And so it's entirely possible that we'll have to attack more than one of these somatic mutations. And as you can see, many of the patients did have more than one, and I'm sure if we look deeper into the sequences, we might find more. So one either has to attack a driver mutation, for which the malignant phenotype is essential, or multiple mutations, so that downregulation of any individual one is not likely to be problematic.
I have 2 questions. I thought it was really interesting, in the case you described, the cholangiocarcinoma, and that the T cells that were reactive against the cancer antigen you described were CD4 cells and not CD8 cells. And so my first question is whether or not you had a chance to look at, if you had a chance to biopsy one of the nodules while the patient was receiving treatment to better understand how those CD4 cells were behaving in vivo. And my second question is whether or not you can comment more generally about the potential of the various T cell subsets in,
Sure.
for anti-tumor therapy.
Yeah. Well, both CD8 and CD4 cells have been shown in animal models to be able to mediate tumor regression. And so we found some CD4s, as you saw in this patient, as well as some patients that seem to have more CD8 related reactivities. What's that?
Steve, I have 2.
There's a question here about NK cells. So I'll make a little bit of a controversial statement, and that is I'm not aware of any instances, with the exception of the allotransplant situation, where NK cells have been shown to have any impact at all on any solid cancer. Probably play a role in the hematologic malignancies, but probably not solid cancers.
I have two related questions, Steve. I'm here.
Oh, okay, go ahead.
I have two related questions. One, do you see any potential advantage in stimulating the reactivity of the T cell that you are transferring adoptively? Or do you think that the response to these mutated antigen is enough, as you showed? That's question number one. And question number two, related to this, do you see any problem with the action of T suppressor cells and the function of these T suppressor cells?
Well, in virtually all adoptive therapy studies, the administration of a vaccine following the adoptive transfer enhances the activity of the cells. And so in animal models, if you give a cell that can recognize the tumor, you can see anti-tumor effects. But if you give a vaccine that specifically stimulates that T cell, then those T cells can expand in vitro and mediate anti-tumor effects. So I think the use of a vaccine can be quite useful, and we haven't done that yet. But since we know what the antigen is, it would be quite possible to synthesize those long peptides and give them in conjunction with the cells. The whole issue of T regulatory cells, myeloid-derived suppressor cells, immunosuppressive cytokines like IL-10, I think these are important issues, which is why it's essential before any T cell transfer to eliminate it's just a brute force elimination of all of the T regulatory cells and myeloid-derived suppressors. It'd be nice to be a little more surgical about it, a little more selective, but we just don't have antibodies that selectively get rid of Tregs or myeloid-derived suppressors. So it's that lymphodepleting chemotherapy that precedes the transfer that's quite critical.
Since you were just discussing all these inhibitory factors, I'm just wondering, as a surgeon, you get to operate on these patients and take out their tumors. Have you noticed that, when we used to make vaccines, we would notice that some tumors would come out like, a baseball, you could hit it to second base and it won't fall apart. And others come out with these soft tumors where you could just run a scalpel on them and all the cells float into suspension. I'm wondering whether you've noticed over time that some of these people who don't respond simply just have such solid tumors that nothing can get in there. Sort of like in the Rakesh chain mindset. I'm wondering what your thoughts are on that.
Well, in vitro, we can grow lymphocytes from virtually any tumor type, probably 90%, maybe 10% of the time. We'll have a tumor from which the lymphocytes cannot extravasate in vitro. And so we don't need the tumor, of course, viable, because we just do exomic sequencing and use that as a target instead of the tumor cell. So as we apply this approach, it may well be that there are some tumors into which these T cells cannot infiltrate, but we'll only learn that by treating a lot more patients.
Thank you.